Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello, and welcome to the Xtalks Life Science podcast. I'm Vera Kovacevic, Editor-in-Chief at Xtalks.com, and this week I'm joined by a very special guest. We have with us today Dr. Sam Lee, Co-Chief Executive Officer and President of Cocrystal Pharma, which is a clinical stage biotech company developing novel therapeutics to meet the growing global need for safe and effective antiviral treatments. They use a unique structure-based drug discovery technology platform and Nobel Prize-winning expertise in their R&D process for antiviral drugs. Dr. Lee has over 25 years of anti-infective drug discovery research experience. Dr. Lee, thank you so much for joining us today.
Thanks for having me. Happy to be here.
Great. So to start us off, we want to get to know you better. We want to get to know Cocrystal Pharma better. Could you begin by sharing a bit about your background and the mission of Cocrystal Pharma in the antiviral space?
Absolutely. My training background is entomology. I've been studying viral replication enzymes. The company, Cocrystal Pharma, was founded by three co-founders, Roger Comber, Gary Wilcox, and myself, back in 2009. And since then, we've been developing broad-spectrum antiviral therapeutics, and we're really excited to talk about influenza and other antiviral drugs today.
Great. So I heard there was recent concern and talk about a new strain of avian flu. Could you explain what this new strain is?
Yes, this is a highly pathogenic avian flu strain classified as influenza A, H5N1. This is a strain that CDC and other research scientists published the sequence. It's a confirmed H5N1 strain.
And why has there been recent talk about this strain in the news?
That's a great question. Avian flus are generally staged with the bird. And typically, in order to infect the human, you have to overcome the host barrier. This is a pretty significant jump in terms of the molecular evolution. The bird virus is designed for infect bird, and in fact that replicated in the bird's intestine rather than lung. And however, through the mutation, they adapt the very specialized mutations, and they then allow to infect the human cells. In this case, in Texas, early this year, the CDC reported that the H5N1 strain, they infected human from Texas farm. It's pretty unusual that the human actually got infected, and they identified also a same strain, H5N1 strain in the milk. Now, recent report showing that Missouri, we have multiple H5N1, clearly showing that possible infection, human-to-human transmission. So they already passed bird flu, in fact, human. Now it's a human-to-human transmission, possibly the first case. So that's a concern.
And the U.S. CDC is currently monitoring the situation?
Yes.
I see. And in light of this new avian flu strain, could you branch out and talk about some of the key benefits, possibly, and the key limitations of current flu vaccines?
Yes. Before we tackle on topic, I just want to talk about pandemic strains. So the pandemic flu strains is actually influenza A strains. I want to differentiate influenza A versus influenza B. Influenza A strains is responsible for seasonal flu, but they are exclusively responsible for pandemic infection. So the current vaccine, 2024-2025 flu vaccines, protects the three different strains: influenza A, H1N1, H3N2, and influenza B. So three strains are the most common strains, that influenza strains, but it doesn't cover influenza H5N1. So avian flus, the one that we detect in Texas is H5N1, but there are other influenza avian strains. H7, for example, H9 is also avian flu. So the current vaccines only cover the existing seasonal influenza strains. It does not cover the H5N1.
I see. And in general, how effective are the current flu vaccines in protecting against seasonal and pandemic flu strains?
Yes, the current flu vaccines are safe, and effective rate is about 60 to 70%. So it's not perfect, but it's been really helpful for protecting seasonal flu. But however, it does not protect the avian flus. And in order to develop the avian flu vaccines, that we'll have to identify, and we have to manufacture and get approval.
I see. And if we talk about the broader space of pandemic preparedness in general, what are the major concerns when it comes to the current level of pandemic preparedness for influenza?
Right. I think every year when we have flu vaccines, the industry and regulators select the strains. So in this case, trivalent, so it covers the three strains. So it really, people don't develop the vaccine against the pandemic strains at this moment for seasonal flu. So when you think about how to prepare, pandemic, preparation, again, there's a gap from existing, vaccine to, the approval in a vaccine. Even if we have a new technology, we can develop the, vaccine quickly. It will take at least several months. It could take more than a year. Then we are entering, different flu season. So there is a significant gap between seasonal approved vaccine and newly emerging pandemic strain.
And could you talk about broad spectrum antivirals? And they seem to be getting attention. How do antivirals work in addressing influenza outbreaks?
This is the question we always think about. What are the gaps here? The vaccines, again, effective rate is 60 to 70%, and this is based on predictions of the circulating strains. People make this cocktail, vaccines. In contrast, the small molecule antiviral therapeutics is a broad spectrum, and regardless the strain, it should inhibit the viral replication. So, for example, Tamiflu, first approved drug, it covers influenza A as well as a pandemic strain and influenza B. A recently approved drug baloxavir, the drug name is Xofluza, and it covers influenza A as well as influenza B. Both drugs are effective. However, both drugs also have issues with the drug resistance. The Tamiflu case, this drug's been used since 1999, so it's an old drug, and so the current circulated influenza virus, roughly 10% of circulating viruses have or may have the Tamiflu resistance. And also newly approved drug, baloxavir, which is targeting the replication enzyme endonuclease of the viral complex. These drugs also introduced brand new drug resistance. That resistance you don't see in naturally occurring influenza A or influenza B strains. So taking these drugs and somewhere around 5 to up to 15% roughly, that patients could develop the drug resistance. So that we need a compounds that has a high barrier to drug resistance is still potent and safe compounds.
So if you were to summarize, what is the current standard of care for influenza, and generally what are the unmet medical needs there?
So Tamiflu is the current standard of care. The unmet medical need is the Tamiflu is you need to treat the patients within 48 hours of infection. It's hard to predict exactly what's the zero infection time because you have a flu symptom. You have to guess. So that window is really critical. Early stage treatment is the ideal for Tamiflu. So I think the compounds we're developing is we want to have, regardless the stage of infection, we want to effectively block the viral replication and reduce the clinical symptoms. That's the game.
I see. And now let's go a bit more into what Cocrystal Pharma is doing, perhaps to address this gap in understanding. So I know that Cocrystal Pharma focuses on targeting the viral replication proteins. Can you tell us how this structure-based drug discovery platform works, and what makes it unique compared to other antiviral discovery approaches?
Absolutely. Our technology is really developed by Roger Kornberg, who received a Nobel Prize, and did a pioneer work on structural biology of the transcription complex. So we're looking at, viral, replication. It's really complex, processes and multiple enzymes. Influenza virus case, 3 different proteins actually coordinate really well, and they produced multiple copies of genomes really rapidly. And that's the mechanism that kills the host cell and reproduce the viral genomes. So for years that we know replication enzymes would be ideal drug target. And the question is, how can we design the drug and targeting the most attractive, drug targets? So influenza cases are 3 different proteins: PB2, PA, which is endonuclease, and PB1, polymerase. These proteins is a big complex, and we recognize that the PB2 protein, which is our first molecule, CC42344. This protein it controls the first step of viral replication. What it does is it recognizes host cell. When virus enters the host human cell, it goes to nucleus. It's the isolated compartment, and it randomly hijacks the host mRNA. So first the step that just virus polymerase, viral polymerase hijack the host mRNA, and then you start generating the primers, and that primer serve as a starting point for polymerase. This is a fundamental mechanism regardless of which virus. But if you look at the seasonal influenza strain versus highly pathogenic influenza strain, for example, Spanish flu, responsible for 1918 Spanish flu, that's the influenza A, H1N1. These pandemic viruses, actual replication rate is extremely fast. In other words, this process is really, really fast. So if you block this first step, PB2, the protein function, we believe that you can control the downstream processes more effectively. So we're working on PB2 protein as well as 2 additional proteins, PA and polymerase inhibitors. So this is the sort of a potential combination therapy candidate as well as a monotherapy drug candidates. So structural biology, the data, it's kind of like a high resolution GPS, if you will. If prior to this, technology, people rely on biochemical, biophysical, or cell-based assays just without knowing the detailed mechanism of action. You just screen the compounds using certain platform, and you isolate the compounds that it has antiviral activity, and that's been pretty good for developing Tamiflu and other compounds. But you don't really know whether your compounds has the activity against the target, for example, PB2 or PA or polymerase. Structural biology work is that we start from the we call drug binding pocket. So we examine before we start the drug discovery work. We carefully examine computationally as well as protein crystal structures. We look at the binding pocket. So typically, drug binding pockets is think of it as a channel. Some channel is a deep cavity. Small molecule come in and finds the space, and they will establish the molecular interaction. Otherwise, you're never gonna see good pharmacological, properties. So this is a process that what we do is we don't use AI, but we do actual computational and experimental data, and to figure it out which drug binding pocket will give us broad spectrum. So before we start the chemistry or design the molecule, we have a pretty high confidence that working on a particular drug binding pocket will give us broad spectrum activity. Now, once we establish this process, then we start improving the potency and continue to confirm the broad spectrum activity. So at the end of the day, we show antiviral activity, we call typically EC50 or IC50, the in vitro potency against the multiple different strains. And that's one important data. But at Cocrystal Pharma, we also provide the additional X-ray data when we overlay all together, whether it's avian flu H5N1 or seasonal flu H3N2. When we look at the drug binding pocket and binding mode, exactly same at atomic level. And I think this is an important scientific foundation, how we confirm the broad spectrum activity. And the second important thing is, as I mentioned to you, drug resistance is a really issue. The virus, RNA viruses, they have the property the polymerase itself, they will spontaneously generate the mutations, and this is the mechanism how they adapt different host infection, as well as that's how they survive. So this is a very important process that viral polymerase has a poor, we call poor fidelity. So continue to introduce the mutation. And our company, the Cocrystal, the platform technology, we will try to predict the potential drug resistance site. And this is experimental as well as computational. So we could actually develop the compounds that suppress, as much as we can suppress the drug resistance. So we call compound with the high barrier to drug resistance. So I think this is a really important aspect, broad spectrum, and as well as a potency and good pharmacology. And finally, the compound with the high barrier to drug resistance. I think this is a really important principle we carry on, and we've been very successful.
Great. And just curious, and for all those science enthusiasts out there, can you talk a little bit more about that Nobel Prize-winning expertise that sort of laid the foundation for a lot of the work that Cocrystal Pharma does? Like, when was that Nobel Prize given, and where was that work done?
Sure. Roger Kornberg and his father, Arthur Kornberg. Arthur Kornberg and Roger Kornberg, both of them Stanford professors. Arthur Kornberg discovered DNA polymerase, received a Nobel Prize in 1959. And then Roger received a Nobel Prize in chemistry in 2006. So before Arthur Kornberg passed, we developed a business plan, again, using this two approaches. One is the enzymology, replication, enzymes, and Arthur did a pioneer work on all of these replication enzymes, which are essential for viral, bacteria, and human replication processes. And so Roger brings this structure biology aspects, and I think that the platform approach, as I mentioned earlier, that before we initiate the chemistry, we carefully exercise the structure biology work and make sure that we have a focused goal, focus the drug target, and offers the broad spectrum activity. I think this is a really profound, or key differentiator from, other companies. In terms of drug resistance, that's another aspect that we have in-house. We have a collection of pretty much all available drug resistance crystal structures. So Tamiflu or Xofluza drug resistance is a famous drug resistance mutation against the Xofluza, baloxavir. That drug is I38T. It's a virus that somehow picked that residue, mutate if you treat the patient with this drug. And in fact, very rapidly, and they isolate this mutation during clinical trial. So so when we look at the X-ray structure of I38T, we understand how the virus could overcome this potent compound. So I think this is putting together not only understand broad spectrum, and I think Roger Kornberg's scientific contributions also beyond potent broad spectrum compounds. Can we develop the pan-viral compounds? And I think that's the topic that I'd like to discuss whenever we have a chance. I think pan-viral viruses, we believe that's the next really generation of antiviral drug.
Great. Yes, I was just going to ask about more of the work that Cocrystal Pharma does on beyond influenza as well. Are there any other viral diseases that the company is currently targeting and looking at?
Yes. So influenza, we stay with the replication enzymes. That's our selected drug target. And during COVID pandemic, we start looking at the protease. It's a completely different drug target, but it is essential for for viral replication. So the protease targets, it's a really unique and interesting. It's a compared to replication complex, it's a one protein rather than multiple protein. And when we carefully look at the viral proteases, we can classify that there are multiple families, but more importantly, for example, coronaviruses, the active site, the drug target, we identified highly conserved drug binding pocket. And that pocket is essentially required for protein function. So when we look at the coronavirus, we're talking about SARS coronavirus 2, MERS, common coronaviruses. We see that architecture, highly conserved architectures everywhere. And so beyond coronavirus, we also expand this knowledge and collect the data. One of the viruses that we found is a norovirus. Norovirus is a completely different RNA viruses. It's responsible for viral gastroenteritis. It's an outbreak. You've maybe seen it in newspaper, cruise ship, military, nursing home, restaurant, foodborne outbreaks, and it's a terrible disease. But regardless, it's a completely different virus. The drug binding pocket that we select, highly conserved. I mean, absolutely, 100%, conservation. So what that means is that, we could develop, using this structural information, beyond coronavirus antivirals, that we can actually come up with the compounds has antiviral activity against, norovirus, for example. And we have a very interesting data in just the same approach. So, now we're gonna call these compounds pan-viral compounds and pan-viral proteases inhibitor. We're extremely excited about this class of compounds because its potential applications are huge. For example, newly emerging the pandemic, RNA viruses, and we don't even, talk about this every day. Borneo virus is one example. West Nile's, dengue virus. All these viruses have this protease that the highly conserved drug binding pocket. So I think this potential application and potential market is huge. So what we're trying to do is develop the molecule, and we do only one phase study, because a phase 1 study is that we demonstrate the safety and tolerability of the healthy volunteers. And once we have this, safety data, we can have a multiple clinical trials and multiple indications, and multiple approval. So it gives you a really huge opportunity.
Yes, I was basically on the heels of that. What excites you the most about the direction of Cocrystal Pharma in the next 2 to 5 years, and in general in the broader field of antiviral drug development as well?
Yes, I think as a drug developer, we want to conduct the clinical trials as soon as we possible. That we want to demonstrate human efficacy and safety, and more importantly, we want to get approved drugs on our pipeline. So next 2 and 5 years, the time window, and then hopefully we'll see first approval drug. And in parallel to what we're doing, we would like to establish additional collaboration with the government agencies as well as the other pharmaceutical companies.
Really good goals and ambitions. And to wrap us up, do you have any message you would like to leave or any advice for those biotech researchers working in the antiviral research space, or just in general to biotech researchers?
I think if you look at the unmet medical need, we, as a scientist and drug developer, we have to always think about better medicine and look for unmet medical need. And this is a really meaningful journey that we have this amazing opportunity to develop the novel drugs, and we're very proud of what we accomplished. But I think that we've learned a lot of things from COVID pandemic. Hopefully, we're not going to see influenza, pandemic influenza. But at this point, as we discussed earlier, if there's no vaccine against the pandemic influenza strain, the current antivirals is working. However, if in fact that avian flu, for example, have a drug resistance, we don't have any other option to think about. So there's always a huge opportunity and unmet medical need. I encourage every scientist to think about, get involved with the biotech company. And I think it's a really meaningful journey.
Great. Thank you very much, Dr. Lee, for sharing those insights with us today as well.
Thanks for having me.
And that's the end of this week's episode of the Xtalks Life Science Podcast. If you like today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we'll see you next week. Bye.
To join the Xtalks community, visit Xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.